Hi, everyone. It's 12 o'clock, so I think we'll get started now. So good afternoon. Welcome to the Clinical Trials Methodology course webinar series. Today's webinar is on study drug formulation and supply issues. But before I introduce our presenter, I'd like to remind you that if you have a question, please use the chat box to the right of your screen, or you can simply unmute yourself. Also, at the end of the webinar today, please take a moment to evaluate the webinar, and I will provide the link at the end of the presentation. So now I'd like to introduce our presenter today. His name is Pat Bolger. Pat is currently the Director of Clinical and Business Affairs at the University of Rochester's Clinical Materials Services Unit, where he provides tactical and strategic support for all stages of clinical trial supply, including project proposals, study kit design, supply chain management, distribution logistics, project management, and regulatory compliance. In addition to practicing hospital pharmacy early in his career, Pat spent over 20 years in the pharmaceutical industry, the majority of which involved developing and managing systems to comply with federal and state regulatory requirements. Pat also led cross-functional product development teams in the planning and execution of strategies to obtain regulatory approval for pharmaceutical products. He received a BS in pharmacy from the University of Florida and an MBA in international business from the Rochester Institute of Technology. So thank you very much, Pat, for taking the time to present today, and I'll hand it over to you.
Great, thank you. Thank you, Joy. Can you hear me okay, Joy?
Yes.
So I'll get going on this. Thank you, everyone, for joining this afternoon. At least it's the afternoon here in Rochester. So as Joy mentioned, what I'm going to cover today are some of the study drug formulation and supply issues associated with clinical trial, supplying a study drug for clinical trials. And the slide that's up now is just a list of some of the topics that I'm going to cover today, and just things to go ahead and think about as you're preparing to consider having a study drug or an intervention component to your clinical trial. And if you have any questions during the course of the presentation, please go ahead and ask during the course of the discussion versus saving them to the end. I think we have enough time to cover them as we go. So some of the topics that we're going to cover today is distinguishing between a single center and multi-center trial, and then get into sourcing of the study drug, all the way back to where it's maybe in its raw material form, or maybe your study drug is further along and closer to being ready for distribution. Also talk about whether or not the study is blinded or open label. And then we'll get into some of the packaging considerations, both the primary and secondary packaging, and we'll touch on those, as well as the labeling, and then also too some of the timeline issues as far as establishing the treatment duration, which is typically well fixed. The enrollment duration, however, is a little bit more of a moving target. And then get into some storage considerations, and then the logistics around distribution and disposition of the study drug at the end of the study. So to start out early on here, when considering a clinical trial or investigator-initiated trial, whether or not it's going to be a single center study or a multi-center study. And single center studies are typically handled within the institution's investigational drug service. So it's actually handled by the pharmacy department within the institution, and it's done on an individual prescription basis, so much more like the practice of pharmacy or a single center study. A multi-center study, and that's what we do here at the Clinical Material Services Unit, is we provide the study drug to multi-center studies and sometimes international studies. But instead of processing pursuant to a prescription, it's much more industry-like, and the regulations that cover our activities are called the Good Manufacturing Practices, or GMPs. So more industry-like, preparing kits in batches versus individual prescriptions, as we see in single-center studies. So a little bit about those GMPs, and I'm going to focus now on the multi-center and the activities that we perform as far as complying with those federal regulations, the good manufacturing practices. And you'll note that there's often a lowercase c that precedes the GMP. And that's just a reminder that it's the current good manufacturing practices, because these are always evolving in order to maintain best practices when it comes to the processing of pharmaceutical products and study drug products. And then really the GMPs are a set of quality standards to ensure that all the processing, labeling are done in accordance with the regs that will maintain the integrity of the product. And one of the overriding philosophies that's present with working within a GMP environment is a done by, check by, a two individual approach to all the regulated activities, where there's a first person who does the actual activity, a second separate person checks to make sure that first person did the activity in accordance with the directions of the procedure. And then again, just pharmacy regulated by the State Board of Pharmacy. So a couple definitions that'll help us get started with the discussion on the formulation of a study drug for clinical trials. The drug substance, or the active pharmaceutical ingredient, and often hear it referred to as API, is the raw material. And then that raw material is then converted into what's called a drug product by definition. And that is basically the finished dosage form. And that's what contains the active ingredient. And interestingly, for clinical trials that are placebo-controlled, the placebo formulation also falls into that definition of drug product, even though it doesn't contain an active pharmaceutical ingredient. So a lot of what we spend our time doing here at the clinical in our operation is involved with sourcing the study drug. And many investigators come to us at various stages of sophistication as far as the readiness of their investigational product for use in a clinical trial. Some already have the study drug lined up to be donated by the pharmaceutical company. Others are all the way at starting from scratch, where they have to try and identify a source of the raw material to get converted into a tablet or capsule that will eventually be used in the trial. So starting with the API, trying to find a source for that if necessary, and then finding a manufacturing organization that'll convert it into tablets and capsules. And then sometimes that manufacturer can also package the product for us, but oftentimes we end up finding yet another manufacturing or packaging operation that will get that into the bottles or blister packs for us. And along that whole path, it's so critical to select vendors who are able to demonstrate their compliance with the GMP requirements. And it really makes our lives a whole lot easier when it comes to resolving any issues that might occur during the course of the trial, and also having some dependable lead times. So once you've got your API, your bulk product, tablets, capsules, and they're packaged, there's also non-drug drug products to take into consideration, being the kit boxes, labels. Sometimes we provide some ancillary supplies, counting trays, oral syringes for liquid products. So there's just a lot of things to keep in mind as early as possible in the planning process. And that's where we get into this keeping in mind as early as possible. The lead time for the supply chain is in terms of months instead of weeks. And as you can imagine, all the stuff we discussed in the previous slide, comes along with a pretty predictable time frame. And from the API again to the components, leaving time for the manufacture of the dosage form, the packaging. And once it's packaged, it typically comes into an operation like ours, and we do the kit assembly, labeling, and distribution. So if we were to go from beginning to end here, sourcing the API, all the way to having material ready for distribution to sites, we're probably looking at least 6 months. So it's again, really, really critical to plan this, have the plan in place as soon as possible. At the bottom here, I've listed another issue that can complicate things from a timeline perspective, and that's if you're sourcing material from an international location, or if there's sites involved that are outside the U.S., just clearing customs can be a weeks-long time requirement in the overall time frame. Now that we've got a nice robust supply chain, and we've got material that's already in bottles and ready to get into kits and out to sites, one of the first things we try and predict, though, is how much we're going to order. And sometimes this seems like an insurmountable task, but it's pretty straightforward, looking at the number of dosages per subject per day, the number of days of dosing, and then eventually the number of subjects. One of the things that's really important to keep in mind as you determine, let's say there's 3 capsules a day for 30 days, and you're going to have maybe monthly visits. It's important to keep in mind that in addition to those 30 days, we need to have that 30-day supply incorporate or cover the visit window. So you may have a visit window for subjects of plus or minus 5 days. So now on top of those 30 days of planned dosing, we need to have an additional 10 days in case a subject comes in 5 days early for one visit and then 5 days late for their next visit. So again, these types of things to keep in mind when you're determining the quantities to order. And then once you've identified what a subject is going to require at each dispensing visit, we also layer in about a 30% overage factor to account for unused kits or kits that go unused at sites. And that's fairly common, and 30% is the standard overage allowance that we use for studies. And why that may seem high and why that is in place is oftentimes if we're doing a study with 3 treatment arms and we're going to have, let's say, a central randomization where a subject could be, they can enroll a subject to any one of those 3 treatment arms. We want to make sure they have those kits available on site to be able to conduct that baseline visit. So at each site, there's going to be one of each of 3 kits. At the end of the study, because we've kept these sites poised to be able to enroll any of 3 subject types, those 3 kits are going to go to waste once the subject meets full enrollment, because we had to be prepared to enroll at any of the sites, any of the treatment arms. So again, that 30% covers things like the planned unused material at sites at the conclusion of the study. And then we identified, as I just mentioned, the quantity of study drug to get your study up and running. And then the next thing we need to take a look at is, depending on the duration of your study, how often are we going to need to refresh the study drug over the course of the study? And again, this is something that we just need to make sure that we've got a plan for in place early on, so that we can keep the study going along smoothly with fresh study drug that is within the expiration date. And we'll get into that in a little more detail. So we've got a supply chain in place now, and we need to keep in mind now whether or not this study is going to be, clinical trial is going to be a blinded versus open label. The majority of our studies that we're handling right now are placebo-controlled, double-blind. However, we do have a couple open label studies, and it's much more straightforward. But when we get into the blinding of material, of study drug, a lot of the studies that we currently handle are looking to repurpose an existing commercial product and study it maybe for a different indication. So if we're going to try and blind an existing commercial tablet or capsule, the most effective and simple way to do this is to have a contract manufacturer over-encapsulate that commercial product into a larger capsule or tablet, and it'll be mixed with a little inactive ingredient to prevent that tablet or capsule from rattling around inside of a larger capsule shell. And then a matching placebo capsule will be prepared with just the inert ingredients, so that now we've got a identical looking dosage form, some active and placebo capsules, one which contains the over-encapsulated commercial product, and then one that's just the diluent. So we're really striving to maintain the blind of the study right from the dosage form all the way up through the packaging into bottles, and then ultimately the labeling of the products. And the packaging is critical as the identical appearance of the capsules. We had a study where a pharmaceutical company agreed to supply the active or placebo tablets, and they were identical, that you couldn't tell them apart. However, when the material arrived at our operation for labeling, the placebo tablets were in 100-count bottles and the active tablets were in 50-count bottles. Well, that's a problem. So we had to do some clever repackaging to get them all into a similar container closure system. So in addition to when blinding, considering a blinded study, in addition to the appearance, um, when it's a liquid product, you end up touching more on these last three bullets: the taste, smell, and texture. So solid dosage forms, tablets, capsules, are pretty easy to blind. Liquid become more of a challenge. Injectable products, even more so. A lot of times with maybe some of these new biologics, we'll get a request where to create a blinded version of a commercial product and injectable product, and a lot of times those auto-inject syringes or vials are very proprietary by design. And so oftentimes for an injectable product where it's nearly impossible to mimic the commercial product, we'll recommend that the study incorporate an unblinded clinician to administer the product to the subject, and then have blinded evaluators, and the subjects, of course, will be blinded. But that if it's too difficult to develop a blinded version of an injectable product, then we would like to see possibly a unblinded clinician administer the drug. So we blinded the product, it's in the bottle, and now the labeling also needs to be blinded. And here's an example of a study we worked on. If you look at the content section of the label, it's an either or statement. It contains either the active or the matching placebo. And that's consistent through the kit box label, which we're looking at right now. This is a label for a kit box that contains 6 bottles of study drug. And then that same either or, that or statement appears in the contents of the bottle label also. And this is just an example of a clinical label that maintains the blind of this placebo-controlled study. So in addition to blinding the capsules, the bottles, the labels, the dosing regimen also needs to be blinded so that subjects can't tell. Subjects and the clinics, the clinical personnel can't tell the difference between the three. In this case example, 3 regimens. So here's a case where we were presented with an issue of 3 dosage levels, 3 treatment arms, placebo, 100 milligram, 300 milligram, and using 100 milligram capsules. The way in which we achieved the blinded dosing regimen was for subjects to take 1 capsule from each of 3 different bottles, bottle 1, bottle 2, bottle 3, daily. So in this placebo treatment arm, 1 capsule from each of the 3 different bottles resulted in 3 placebo capsules. The 100 milligram kits, they had 1 active bottle and 2 placebo bottles. So they again took 1 from each of the 3 bottles and achieved their 100 milligram daily dose. And likewise, the 300 milligram group with 3 of the active bottles were taking the same dosing regimen, 1 capsule from each of the 3 different bottles, and achieved these 3 different dosages by having an identical dosing regimen. Sometimes these studies have a titration phase, and maintaining the blind during that titration is also important. And in this example, we're looking at, again, a 3 treatment arm study, 150, 300 of placebo. And in order for the subjects to reach their highest tolerated dose, they wanted to start out at 150 milligrams and then see if they could step them up to tolerate a 300 milligram daily dose. So week 1, they're taking 1 capsule a day from 1 capsule a day from bottle A. And then at week 2, we introduce bottle B, and they start taking 1 capsule a day from bottle B in addition to the bottle A. So you can see the 150 milligram treatment arm, when they started taking bottle B, it was just a placebo, and they maxed out at 150. The 300 milligrams bottle B contained 150 milligram capsules, and they got all the way up to 300 milligrams. So again, things to keep in mind with the dosing regimen in order to maintain a titration phase of the study. And then we have the studies where they want to allow for a dose reduction. And again, important to maintain the blind so that independent of what dosing arm you're in, if you're going to step down, we need to make sure that, especially the clinic personnel, when they step down, they need to be stepping down the same as far as reducing dose from the same bottle in order not to sort of tip our hat as far as what's in each of the bottles or what treatment arm a subject might belong to. So in this same example, when subjects in the 150 milligram treatment arm, let's say, stop tolerating the current dose, then we're going to ask them to stop taking a capsule from bottle A, and that'll get them down to just the placebo capsule daily. The 300 milligram treatment arm will also stop taking from bottle A, and that'll get them down to the 150. If we had them stop taking from bottle B, it wouldn't have worked for the 150 milligram treatment arm because they would have discontinued their placebo capsule and still been at the 150 milligram level. So just things to keep in mind as far as configuring your kits and allowing for either dose titration or dose reduction or both. So some things also to keep in mind as you're working on the formulations for your clinical trial. The size of the capsule or tablet is important, and one of the challenges we bump into when we blind commercial products with that over-encapsulation, sometimes that larger capsule can get to be a significant size and become difficult to swallow. And as you can imagine, you don't want to allow any of these over-encapsulated products to be opened and sprinkled on food, because that would reveal that there's an over-encapsulated commercial product inside that capsule. So we want to make sure that over-encapsulated product is taken in an intact capsule. So size of the capsule is something to keep in mind, and then packaging and labeling. The child-resistant products work well for child-resistant, but they can be adult-resistant. So something just to keep that in mind with the population of your study. And then also, it's tempting to put as much information as possible on the label, but we really try and limit it to the minimum requirement, just so that we can keep the font size at a level where they can read the instructions. So again, things to keep in mind from a compliance standpoint with labeling and dosage form. I touched a little bit and just mentioned primary packaging. That, if you heard the term primary packaging, it just refers to the bottle or blister pack that the capsules or tablets will go into. It's the immediate container that maintains the integrity of the product. And here's an example of some different types of primary packaging: glass, plastic, blisters, and then for topicals, tube sprays or pumps. So once we have the material into the its primary package, there's testing done to ensure that the product is the integrity of the product hasn't been somehow impacted by the primary packaging. So there's testing that's done at the time of the primary packaging or at the time of manufacture, and then there's also testing done over the course of several months or years, and that's called stability testing. And so the stability test is in place, and it's even in place in that example I keep referring to of over-encapsulated commercial product. The commercial product that we purchase from the wholesale level will have a fixed expiration date. When we combine it, or when we over-encapsulate it and combine it with some inert ingredients, then we also have to do stability testing on that, because that fixed expiration date of the commercial product applied only to the original container that contained the commercial product. So once you open a bottle of ibuprofen that you purchased at the pharmacy, then and you decide to over-encapsulate that original expiration date on the ibuprofen doesn't apply anymore. We have to do additional testing to make sure our over-encapsulation didn't somehow change the integrity of the product. So at the bottom here, you'll see different stability programs and the different conditions of stability programs. And the ambient conditions or room temperature is what the long-term stability programs are run at. The accelerated conditions we do because when we first develop a product, whether it's for investigational use or even in commercial use, we don't have real-time stability data that would say, okay, this product is going to be okay at room temperature for this many months. We can't wait, we don't always have the luxury to wait 6, 12 months to say, all right, we know this product is going to be good for at least that long. What we'll do is put that product up on accelerated stability conditions and really stress the heck out of it with high humidity and high temperature. And if it's behaving okay and the tests are within specifications at accelerated conditions, we have a pretty good feeling that it's going to behave well at room temperature for 12 plus months. You'll hear a couple different terms used to describe the shelf life of clinical trial materials. There's a retest date, and this is, I guess, the best way to look at this is sort of a rolling expiration date. It's a date where there isn't a fixed expiration date in place yet, and this can apply to the active pharmaceutical ingredient or the drug product, where it's tested at certain intervals to ensure that it's good for at least that long, at that period of time, and then it's tested again. So it's I guess a rolling expiration date, and this is something that is in place when you don't have a fixed expiration date available for the product. And here we are at expiration dates, and that typically applies to marketed products. And the way pharmaceutical companies are able to arrive at that is they have a significant body of data, stability data, that indicates that the product at room temperature or whatever the desired, the required storage condition is good for at least as long as what they're putting on the label. And they usually allow themselves a little bit of leeway here that commercial expiration dates are usually 2 to 3 years, but it's very likely that the product will be good for 4 to 5 years. But the 2 to 3 is errs on the safe side. And as I mentioned, once that original container is opened, the original expiration date is no longer doesn't apply to the full shelf life that would have been stated on the label. Probably good for the indicated amount of time, but that stability data is really on that original unopened container. Here are just a few examples of some analytical testing that might be done at the time of manufacture or during while on stability. And disintegration, dissolution is just a method of measuring the rate at which the drug comes out of the off the dosage form or out of the dosage form in a dissolution bath. And then the assay would measure the amount of active ingredient that can be recovered following dissolution to make sure that if there's 200 milligrams of ibuprofen formulated into a tablet, we're able to assay that and determine that that there is plus or minus 200 milligrams that was measured to be in that tablet. And then some micro testing, make sure there's no bugs in it, and then preservative effectiveness tests for multi-use oral liquids and vials, just to ensure that the system that's in place to minimize contamination is working the way we would expect as that multi-dose container is used over and over. Now moving into some of the timeline considerations as you're developing the plan for your investigational product. And one of the most critical pieces of information and things that's oftentimes most difficult to nail down at the beginning of the study is the enrollment duration. And that really drives how often we're going to need to refresh the study drug over the course of the study. So if it's an overall 3 or 4 year study, excuse me, and we are pretty confident that the material coming out of the manufacturing and packaging process is going to be good for 18 to 24 months, we're going to need to refresh that study drug over the duration of the study just to make sure that those original kits are going to become unusable, a year or two into the study. So we're going to need to have fresh kits available so that the study can continue to run smoothly. When we over-encapsulate commercial product, it typically starts out with 2 to 3 years of expiration or shelf life. And then by the time we get it into the over-encapsulation and packaging process, we're going to chew up 4 to 6 months. So that product that we buy from the wholesaler with 24 months of dating is only going to have 18 months remaining when we begin distribution out the site. So it's really critical that we keep those sort of high-level timelines in place because this is often, well, not often, all the time, the biggest challenge for us when managing the supply chain or the study drug for clinical trials. So, and not to beat a a dead horse here, but the enrollment duration, if it's slower than expected, and it often is, it really complicates the expiration, managing the shelf life. We're right now handling between 15 and 20 studies, and I would say 90% of them are experiencing slower than expected enrollment. And it's just very, very optimistic when the studies are in the planning phases and getting ready to be submitting that application for funding, or the grant application. And there's often a lot of pressure, as you all know, in the first patient in. We got to be able to celebrate that milestone, and a lot of times there's a significant amount of effort to get that first patient in at maybe the investigators of their institution site, and then it'll be a little bit of a lag before the other sites come on board. So we may get that first patient in today, but that second site might not become operational for another 3 months. And yeah, we met the first patient in milestone, but now that second site came along 3 months later and just chewed up those 18 months, three of those 18 months of remaining shelf life. So we often try to encourage studies to not start that shelf life clock ticking until they expect to have as many sites as possible up and enrolling. Sticking with the sort of the timeline, study timeline theme here, important to try and standardize when we build these, develop our quotes and construct these kits. We like to maintain as few as possible different variations of kits. Let's say for a study that's going to have a 3-year study, and they're going to have dispensing visits every 6 months. We'll ideally build active and placebo kits that are 6-month kits, and sometimes that 6-month kit, you might look at that first 6-month period and say, oh no, we've got a baseline visit and a 2-month visit, and then we fall into 6-month kit. What we'll recommend then is let's stay with that 6-bottle 6-month kit, but let's only give out 2 bottles at baseline, and then the remaining 4 bottles at the 2-month visit, and then we'll get into that 6-month kit cycle. So we really try and standardize the kits as much as possible. Another thing here in the second bullet point that we sometimes bump into, that early on in the study we like to see our patient, maybe that visit window is plus or minus 7 days, and then in the out years it might be plus or minus 14 days. It's just something to keep in mind, that we will need to build that kit to accommodate that. If we're going to have a single kit, 6-month kit that's going to last the entire study, or be usable for the entire study, then we need to make sure that we accommodate sort of the worst case scenario in terms of the visit window and the longest visit window that could possibly happen. This third bullet point, this is a bit of a little case, kind of something we learned, a little bit of a case study. We had a study that was going to have dispensing visits every 3 months, and the fallback here was, okay, those 3 months are, we're going to have 3 4-week kits in those 3-month period. And that sounded great when it started, but if you look at the calendar, there's 13 weeks every 3 months. So the best laid plans there came up a week short. So again, something to keep in mind here is that when designing these kits, really take a careful look at how many days of dosing you're going to support and how many visit window days you need to support in that particular dispensing. Storage conditions, we've been talking mostly about the room temperature conditions, but there are studies where we have been involved with frozen and refrigerated. Something that the contract manufacturers can certainly handle, and ourselves, we're set up to handle this. Down there in red at the bottom, one of the things that we often have to be more sensitive to is the fact that clinical sites, may not have a huge capacity for frozen and refrigerated storage. So we just kind of need to keep that in mind, and that applies to room temperature storage too. Some of these sites have really limited storage capability when we want to send out larger than fairly bulky drug products. When we do have a temperature-sensitive investigational product, we will include a temperature monitoring device in the shipper when we send it out to sites. And this is an example of one we commonly use called a temp tail. And when we pack the shipment for on its way to the site, we'll hit the green button to start the recording, and when the site receives it, they'll hit the red button to stop recording temperatures. Then they connect the USB port to their computer, and it'll generate a nice little PDF of the blue line here indicates the temperature that was experienced while in transit. So it's a way to ensure that the integrity of the product and confirm the integrity of the product while in transit out to sites, and that's a very common practice for temperature-sensitive shipments. Now that we've got product out to the sites at the proper temperature and it's all blinded and the kits are perfect to cover the dosing days plus the visit window, we're going to need to perform some accountability of the drug product. And we have that accountability in place throughout the supply chain. But once we get it out to sites, the SOPs in place at the sites need to make sure that they have that accountability piece covered. And a lot of times that accountability piece sort of doubles as a bit of a compliance piece too. So from a drug accountability perspective, here's an example of a form where we have by title here, a drug compliance accountability log. And in this particular form, and these are often customized to a particular trial, but in this form here, kind of a checkbook type of an approach, but in the left-hand column, there's the 8 different visits that are identified. In this case, there was the baseline visit, and then there was visits 2, 3, 4, 5, and 6. There were also a couple telephone visits here. And in this particular study, although the subject didn't come in to the clinic, the sites mailed them a 6-month kit based on a telephone visit. So we needed space to record the kit numbers that were dispensed at each of those visits, and then tracking who dispensed, the date of dispensing, the date of return, how much was returned. So this is the type of form that is required for accountability, but it can also be a bit of a compliance tool also. And at the conclusion of the study, we need to decide, now we've got study drug, that's unused or sitting around at the sites or sitting in a central inventory, and we just need to have a plan for, are the sites going to, and it's most economical if the sites can destroy unused study drug, and according to their local institutional policy. But either plan for knowing that they're going to destroy on site, they're going to return it to us, maybe send it off to a third party, and then also for the central inventory, what we have here that we never dispense to sites. We'll think about whether we want to return those to the sponsor or again send the product to a third party for destruction. Everything handled in an environmentally sound way. We do bump into issues most commonly in California where the environmental restrictions prevent their institution from destroying the drug product. So we are obligated to take that back and see that it's destroyed according to environmental regs once it's here. I think that disposition of study drug is my last slide, and that concludes the presentation. I'd be happy to field any questions that may have come up along the way. Still there?
Yes, please. If you have a question, please use the chat box or unmute yourself to the right. You can turn your audio on. Are there any questions, comments? So there is a question, Pat. Do you see that in the chat box? From Brian Trummer saying, sorry, did I miss about evaluation of taste? Saw it on the slide.
Oh, I'm sorry, evaluation of?
His taste, he saw it on a slide.
Yes, yep. So we have had, and it's typically with liquid products, where we do need to mimic the taste. Some drugs are inherently bitter or sweet, and we have to sometimes mix in an ingredient that will help mimic that taste. We've had a study where we were looking at an elixir in infants, and elixirs by definition contain alcohol, and we had to come up with a formulation because it was unethical to expose, for the placebo arm, unethical to expose infants to unnecessarily to ethanol, to come up with something that would mimic the presence of ethanol in an orally administered product. So right down to color, taste, everything, texture is something that really comes into play with especially oral liquids.
Okay, there's another question from Robin Conway. She said to please comment further on products lasting longer than the expiration date.
Oh boy. Yeah, thank you, Robin. We typically have the expiration date as the hard stop, and of we not to be able to use drug beyond that hard stop. There have been some isolated instances where, with the approval from the regulators, that the drug, as long as it continues to be tested, may be used beyond that commercial shelf life. But that's really the exception. I'm almost hesitant to mention it because it is it's a very unusual situation that requires a special blessing from the regulators and also the IRB, that there, if we were to try and use the product beyond its commercial shelf life, we know from a practical purposes, that my pharmacy hat, that the day that the commercial product, the day after the expiration date of the commercial product, it probably just doesn't all deteriorate and become useless. For personal use, it's one thing to take your ibuprofen for 6 months beyond the commercial expiration date, but in a regulated situation of a clinical trial, if we're going to do that, and I would strongly recommend that we don't, but if it does come up, we need to make sure all our ducks are in a row and then the approvals are in place to be able to extend that for a short period of time. And often what causes that to come into play is lower than expected enrollment, limited study budget. We want to eke out 3 more months of the study, but we don't want to manufacture, we don't have in our budget to manufacture any fresh study, any fresh investigational products. So we are backed into a corner if we even have to consider that option.
Okay, great. So the next question is, for a 1-year trial, how often would you resupply study drugs to subjects? Every 3 months, perhaps?
Yes, I would. I would keep it at 3 months, 6 months. One of the things, you might think, well, we can just give them a single 12-month kit, and that would certainly sound simple, but that means that if we have that kit that's ready for shipment out to sites, and there's 18-month shelf life remaining on that kit, those 12-month kits, those kits that support 12-month dosing, only have 6 months of useful dispensing life because they've got to cover that full 12-month dosing. So I would definitely recommend that we go with something like 3 or 6 months at the most as far as the duration of dosing which that kit would support.
Okay. And then the next question is, the accountability log template is patient-specific, confirms drug kits dispensed to each subject. So is there also an accountability log to confirm the site received drug from
great
you
question, yes.
and are at the site pharmacy ready for delivery?
Then that's a great question. And there is, that was the subject level accountability. There is also a site level accountability where, you would see a very similar looking form, but it would be tracking kits in and out of the institution without, the specific subject activity. And while we're on that topic of the investigational pharmacy, and I didn't touch on it, but there's a couple different systems in place as far as the controlling the dispensing and the inventory of investigational product. There's automated system called an interactive web response system that is very much an automated version that there's a central database and system that we interact with to tell it what kit numbers have been sent to a site. And then when it comes time to randomize, the site personnel log into that system, and the system knows which kits are active and placebo based on what we've told it. But the blinded site personnel, the system will just tell them dispense number kit 123, and the system will know whether or not it's the appropriate kit for that to match that the treatment arm to which that subject's been randomized. The other way to do this is to have a subject where kits are identify the subject's enrollment number or randomization number, and those kits are really without an IWRS that allows the flexibility of a unique kit to go in the direction of any treatment arm. These kits are actually prepared and sort of dedicated to a placebo subject or active subject. So the IWRS comes with additional expense, but it also comes with a lot of flexibility.
Okay, and the next question is, how much per tablet does it generally cost to create a placebo that tastes and looks similar to a commercial tablet medicine?
I'm going to have to defer. I'll go back and check some budgets. We don't do the dosage form manufacturing piece here. We don't make the capsules and tablets. We work with contract manufacturers that do exactly that, and I'd have to go back and look at some of their budgets to see what the per dosage form piece is. And just to caution too, it's not just the manufacture of that placebo capsule and then the packaging of that placebo capsule. The placebos also undergo testing to ensure that there's no microcontamination, and that the placebo capsules are actually tested for the absence of active ingredient to make sure that they're actually placebo capsules. So the analytical testing piece can get to be quite expensive. It doesn't take long for analytical testing, developing the method and validating it to get into six figures.
Okay, and then there was a question if the slide set would be available, and would you mind us posting it to our website along with the videos to this webinar?
Nope, not. Okay. No, that'd be great.
Awesome. And then the last question is, what insights could you provide regarding patient medication compliance technologies that have been developed or are on the horizon?
We have experience with one trial that used those smart caps that actually record when the bottle is opened, and it captures the opening and closure, the timing and the frequency of how many times that bottle has been opened, and those work quite nicely. This was a PK study, so it was really important to know the time of the dose, the subject dose themselves. They are also dependent on the subject after opening the bottle to actually taking the capsule or tablet. So they come with some possible shortfalls, but I've seen them work quite well when it was really critical to the study to know exactly when the study drug was administered. So I think that they're coming and it's improving.
Great. Are there any other questions out there? That was the last in the chat box. Or any comments? There was a comment, Pat, that this was a very practical and helpful overview. So thank you. Appreciate that.
Well, good. No, I'm glad that was helpful for him.
Any other comments or questions? Okay, I guess we will end here. And thank you, Pat. I really appreciate your presentation and thoughts and comments on everything. So thank you. And I just wanted to remind everybody to evaluate the webinar. I posted the link on the chat box here. So thank you, everyone. And we will be sending out another webinar in September. The next webinar is September 10th on good clinical practice. So thank you, everyone, and have a great day. Bye.
Yep, thank you.
Bye.